Hi, today I am making a detailed presentation of my work on topology-aware generative adversarial network for joint prediction of multiple brain graphs from a single brain graph. This work has been done at Basira Lab by myself, Ala Bessadok, and supervised by Dr. Mohamed Ali Mahjoub and Dr. Islam Rekik. Nowadays, brain graph or connectome is an important tool to characterize the abnormal changes in the human brain. It is conventionally constructed from medical modalities such as magnetic resonance imaging or MRI. However, existing multimodal medical datasets that contain a variety of neuroimaging modalities such as MRI or computed tomography or positron emission tomography usually have incomplete observations due to the high acquisition cost and processing time of multimodal scans. So there is a need for a brain graph synthesis solution. To address this issue, a recent brain graph synthesis work proposed a geometric deep learning framework for a one-target brain graph prediction from a source brain graph. However, it is not effective for jointly predicting multiple target brain graphs from a single source graph. For example, if we have two target graphs to predict, we need to train two distinct GDL models for each target domain. So the question we aim to answer in this work is how can we design a GDL framework for a multi-target brain graph prediction task where we train a single model for all target domains. Existing multimodal image synthesis studies are categorized into one-target and multi-target prediction approaches. In the first category, some studies make use of a single source data available to predict the target data, while some other studies used multiple source data for one-target modality prediction. Since such one-target prediction frameworks are incapable for jointly predicting multiple target modalities using a single learning model, recent studies focused on multi-target prediction tasks such as Huang and co-authors, which proposed an adversarial autoencoder framework to predict three target MRI, T1, T2, and FLAIR from a single source T1 MRI scan. Another recent study also adopted a similar architecture to the adversarial autoencoder for computer vision tasks. However, these frameworks are designed for image synthesis, so they cannot be generalized to geometric data types, such as brain graphs. Concerning the graph synthesis task, we have identified a single brain graph prediction work, while many frameworks were designed for the molecular graph synthesis. HADA, which is an adversarial domain alignment framework, was proposed to predict a single target brain graph from a source one. Besides its ineffectiveness for multi-target prediction, as we mentioned before, it is not designed in an end-to-end learning model. It mainly decomposes the model into separate parts that do not co-learn, which leads to relatively high accumulated errors across the learning steps. On the other hand, Su and co-authors proposed a variational recurrent neural network to sequentially generate subgraphs in order to generate the whole molecular graph. However, these graph synthesis works only learn the global graph structure, like the number of nodes and edges weights, and overlook the learning of the local graph structure, like how central a node is in the graph. By overlooking the learning of the local graph structure, one cannot capture which ROIs would be most effective for early diagnosing a disease. To overcome all these limitations, we introduce MultiGraphGAN, the first attempt to jointly predict multiple brain graphs from a single brain graph. First, we design a graph adversarial autoencoder with an end-to-end learning fashion, which is generic so it can be applicable for both one-target and multi-target graph prediction tasks. Second, we design a cluster-specific decoder to accurately generate the target graphs. This handles the mode collapse problem of GAN, where the decoder produces data that mimic a few modes of the target domain. Finally, we introduce a topology-aware adversarial loss function to enforce the generated graphs to retain a centrality score of each node in the original target brain graph. This preserves both global and local topological properties when predicting the target graphs. Here I give an overview of the main steps of our proposed method. In the first step, we represent our multi-view brain graphs by a single graph population, which models the relationship between training samples in a given domain. In the second step, we learn the source graph embeddings and cluster the resulting embeddings into homogeneous groups. In the last step, we train a set of cluster-specific generators to predict the multi-target graphs, which are regularized using a single discriminator. Let's start with the first step. Each subject is represented by one source brain graph and m target graphs, where each graph is encoded in a symmetric matrix whose elements measure the similarity between two brain regions. So we vectorize the off-diagonal upper diagonal part of each matrix to create a feature vector F that encodes the connectivity features of subjects in a specific domain. Next, we stack those features vertically to get a feature matrix for each domain. We recall here that we aim in the following step to cluster the source brain graphs of the training subjects. To do this, we propose to model the relationship between these subjects. So, we learn a sample similarity matrix S for each domain using multi-kernel manifold learning algorithm, or MKML, which efficiently fits the statistical distribution of the data by learning multiple kernels. Following the construction of the feature matrices and the learning of the sample similarity matrices, we represent our set of graph populations as G, where nodes denote subjects represented by their brain features and the edges represent the pairwise similarity between subjects. Now, as we created the graph populations, we will use them in the following steps. Essentially, in the second step, we will learn the graph embeddings of the source population. Our MultiGraphGAN is mainly a graph adversarial autoencoder comprising an encoder E and a set of domain-specific decoders, also called generators, and regularized by discriminator D. First, we learn the source graph embeddings of the population using an encoder E. Second, we decode the resulting embeddings using a set of decoder, each dedicated to a specific target domain, and the results of the generators represent the predicted target graphs. And we optimize the learning process by backpropagating the loss of the discriminator and the joint loss of the generators. So we define our encoder E as a graph convolutional network, or GCN, taking as input the two characteristics of the source graph population, the source feature matrix Fs and the learned sample similarity matrix Ss, and producing the embeddings of the source graphs encoded in the Z matrix. We also use the same encoder architecture to build our decoders. They mainly take as input the Z matrix as a feature matrix and the sample similarity matrix learned for a specific domain. However, in practice, such GAN generators do not handle the heterogeneity of the input data distribution. They generally produce graphs that match a few samples of the target domain. To solve this problem, we propose to first cluster the source graph embedding Z into homogeneous clusters. We also use MKML for clustering, since it outperformed state-of-the-art clustering methods when dealing with biological datasets. By doing so, we are enforcing the generators to learn from homogeneous distributions, so we are reducing the generator's risk to match a few samples of the target domain. To decode the resulting clustered embeddings, we proposed in the next and final step a set of cluster-specific generators. Basically, for each of the C clusters, we build M generators, where each aims to predict the target graph of a single domain. So, for example, these two ping generators aim to produce the graphs in the target domain 1 and the target domain M of the subjects belonging to the same ping cluster. In that way, we are building C generators for a target domain instead of assigning a single generator, where each produces a graph approximating the target data distribution of a specific cluster. More specifically, each generator takes two inputs, the source embedding Z of a specific cluster and the sample similarity matrix S learned using the target graphs in the same domain of that specific cluster. All of the predicted target graphs are optimized using the discriminator D, which we define it as a GCN model. Its main role is enforcing the generated target graphs to match the ground truth target distribution of a specific domain, which is achieved using three loss terms computed for each of the C cluster-specific generators. The first one is the adversarial loss, which measures the realness of the generated graphs by computing the Wasserstein distance among all domains. This distance stabilizes the training process of our generative models. The second loss term is the graph domain classification loss. Here we define a binary classifier DC on top of our discriminator D, which classifies the predicted graphs as 0 and the real target graphs as 1. So we compute the mean squared loss between the predicted labels and the ground truth label of our distribution. This loss term enforces our cluster-specific generators to accurately produce graphs that are properly classified by the discriminator to the specific target domain. The last loss term we included in the discriminator loss function is the gradient penalty, which is adopted by the MWGAN framework since it improves the training stability of our model. After computing these three terms, we compute the final loss of the discriminator, which sum up the loss terms of the C clusters, where the lambda GDC and the lambda GP are hyperparameters to be tuned. Now, as we defined the loss function of the discriminator, let's see how we defined the loss function of the cluster-specific generators. We recall here that the brain graphs have unique topological properties for biological connectivities that should be preserved when predicting the target brain graphs. For example, in these ground truth graphs, if the yellow ROI is considered as a central node for the subject S1, which means the most effective node in passing brain signals, this does not mean that it is also a central ROI for the brain of the subject Sn. To preserve both local and global topological properties of the target graphs, we propose a topological loss function that is made up of local and global topology losses. Given a centrality measure such as closeness centrality, which quantifies the closeness of a node to all other nodes in the graph, our proposed local topology loss is defined as the absolute difference between the real and predicted centrality scores of each node in the target graph. The second part of our topological loss function is the global topology loss, which is computed as the absolute difference between the real and predicted feature matrices. This enforces our cluster-specific generators to preserve the relationship between the ROIs in terms of number of edges and their weights. Basically, we compute these losses for each target domain in a specific cluster, then we sum up the results of the M target domains. To guarantee that the source brain graphs can be also generated from the predicted target graphs, we propose a graph reconstruction loss function which is similar to the topological loss function. We input to the generator the predicted target feature matrix and its corresponding similarity matrix. Then the generator produces the reconstructed source graphs which preserve the global and local topological properties of the source domain. To further improve the quality of the predicted target graphs, we integrate the information maximization loss term introduced in the MWGAN paper. Since the target domains are correlated, this term enforces the generators to correlate the predicted graphs with a specific target domain. So that was the loss function of the cluster-specific generators, where these three lambda represent hyperparameters to control the relative importance of topological loss, graph reconstruction loss, and information maximization loss. In our experiments, we used a set of 310 structural T1 weighted MRI data extracted from the EBI dataset. Each subject has 6 morphological brain graphs or MBG with 3 graphs for each hemisphere: the maximum principal curvature, mean of average curvature, and mean so-called depth. We consider the maximum principal curvature of the left hemisphere as the source graph used for 5 target graphs prediction. For evaluation, we trained our model on 19% of the dataset and tested on 10%. And we also used grid search to determine the best parameters for MKML and our graph autoencoder. We compared our MultiGraphGAN against 2 baseline methods and its 3 ablated versions. The first baseline method has the same architecture of MWGAN that we adapted to graph data types, but we neither included the clustering step nor our proposed topological loss function. The second one is a variant of the first method where we add the MKML clustering of the source graph embeddings. And the 3 ablated versions are mainly our MultiGraphGAN models learned using 3 different centrality measures: closeness, betweenness, and eigenvector centrality. We report for each of these 5 methods the Pearson correlation coefficient between the ground truth and predicted graphs, and the mean absolute error between the ground truth centrality scores and the predicted ones. We can see that with both evaluation metrics, our MultiGraphGAN framework outperformed the comparison methods. This demonstrates that our proposed cluster-specific generators improve the MultiTarget graph prediction task by avoiding the mode collapse problem. It also shows the advantage of our topological constraint for optimally learning the target graph structure. In this work, we introduced the first geometric deep learning framework with an end-to-end learning manner for multi-target brain graph synthesis from a source graph. Our MultiGraphGAN has 2 main strengths: the clustering of the learned source graph embeddings, then training a set of cluster-specific generators which synergetically predict the target brain graphs, and the topological loss function, including a centrality measure which enforces the generators to preserve local and global topology of the original target graphs. Our MultiGraphGAN showed that eigenvector centrality and betweenness centrality have much impact on identifying the most influential node rather than closeness centrality. We aim in the future to extend our framework to predict multi-target time-dependent brain graphs, and we will evaluate it using a larger dataset including functional and structural brain graphs. If you want to use our code, please check our 3 minutes long video on our Basira YouTube channel about how to run our Python code on your own data. You can find our GitHub link in the description below.